Hey everybody, in this episode we're going to deal with what to do with the core of the paper, which really is the experimental part, the materials and methods and the results section, right? That's kind of the heart and soul of the paper. The intro is interesting, conclusions are interesting, the abstract's interesting, title's interesting. None of that adds up to anything if you don't have a bunch of experiments to report on, something new, right? So I'm going to give you just a quick overview at this point of the materials and methods and then of the results section of a paper. And then in later episodes, we're going to go into much greater depth into what to do with both of these because they're so important. So your materials and methods section are just what the name implies. This is the actual, practically, protocols. Now, technically speaking, a protocol is going to be step 1, step 2, step 3, step 4, and they're not quite going to get it to that level, but they will give you all the information you need to be able to actually lay it out, step 1, step 2, step 3, step 4. So if your purpose for reading this paper is to learn a new method or improve upon a method you're already doing, you're going to want to pay very close attention to the materials and methods sections. And you may even want to kind of map it out. You may want to actually put together a workflow, step 1, step 2. You may even want to diagram it so you can visually see every step involved. And oh yeah, before I do this, I'm going to have to do this, and so on. But a good materials and methods section is going to have all the information you need, at least in theory, to completely reproduce the experiments, and if all went well, to reproduce the results that were obtained. One way to approach the materials and methods, if your purpose is not specifically methodological, you're not reading the paper to figure out a precise method for something, I'd recommend you don't skip the materials and methods, but skim them. Okay, look over them, look at the major headings, description of the study site, glance through the information so that you have a general map, a general idea of the major pieces that were involved. So here we can see there's a study site. We already knew from the title there was a field site involved, right? These urban coastal wetlands. So if it's really important to us, we can see what the study site is all about. If that's not real important, we can move on. We glance through, we see Tijuana, Mexico. Okay, that comes into play. We see surface sediments. We see the word estuary. All right, so we're talking about some kind of a coastal estuarine environment. We're looking at the mud, not the water. Got the basics there. Antibiotic abbreviations, we'll come back to those. We know they're there. We'll come back to them when we're looking at results. If we see an abbreviation that we don't recognize, we know where to find it now. Now we see endogenous versus exogenous plasmid capture. Sounds like it might be important to figure out the two different ways that these plasmids were captured. Endogenous meaning coming from within, exogenous coming from the outside. What are they getting at with that when it comes to plasmid capture? Better skim through both of these to discover what the authors mean, because chances are, I mean, if they made enough of an effort to delineate these two in the methods, it's probably going to be delineated in the results as well, probably going to be important. Mobilization experiments, hey, we're plasmid biologists, that's why we're reading this paper, and so we know what mobilization means. And so we can look and see what they did in order to try to get conjugation to take place. Antimicrobial susceptibility testing, we can see they give a real short paragraph suggesting that they use standard techniques. That's really helpful in a methods section when standard techniques are used and it's not a brand new experimental design. Often they can simply reference those standard techniques. And if it's really essential for you to know those techniques, you can go look it up in the reference. But otherwise, you can see here, very short paragraph, disk diffusion method according to CLSI protocols and standards. Here's the link. Okay, great. They use standard methods. And if it's something you're already familiar with, awesome, you move on. If it's brand new to you and you're like, hey, what is this all about? You're going to have to go look that up and start reading. Looks like they determined plasmid copy number. It's an important part of plasmid biology, and they used qPCR to do that. Do I need the absolute details? No. I know what plasmid copy number is. I know what qPCR is. Great. I'll be able to interpret the results when that comes around. Am I interested in how they sequenced the plasmid genome? Nah, maybe I'm not. Maybe all I care about is the fact that they sequenced it, they annotated it, and they analyzed it. And if there's anything really novel in the results, regarding an approach they used, I may go back and find the software that they used for annotation analysis or something along those lines. And the nucleotide accession numbers are here. So let's say that I'm a plasmid biologist and I really want to actually analyze some portion of the sequence data myself. The A's and T's and C's and G's are not going to be recorded in these results. It's just way too much raw data. But I can see here that there are accession numbers in GenBank, and so I can go find the plasmid I'm interested in. I can download that sequence and run my own analyses that I'm interested in if I want to do that. Okay, so I've just worked through that methods and materials. I've got a pretty good general idea of the techniques used and generally what steps they use to capture and then ultimately analyze these plasmids. And so now it's time to jump into the results. You're going to spend a lot of time on the results on the paper because this is really the meat of it. If there's only one bit of this whole paper you're interested in, I'd recommend you jump to it. Let's say you really just wanted to see these plasmid maps because you've got some question you're asking in your own research about whether BLATEM1B is always found next to the FLOR gene. I don't know, something like that. You may want to jump straight to the maps then, or straight to whatever data it is you're most interested in. If you're interested in the entire study, then work through it methodically. Read through the text, and anytime it tells you parenthetically to refer to a figure or table, end that sentence, go look at the figure and table, and come back to the text and pick up with it again. You're going to be going back and forth between the text and the figures and tables, back and forth, because they're going to be sort of working off of each other. Subheadings can be really helpful. So here's a section talking about one of those 4 plasmids that they named in their title. There's a table there, lots of information in words that may not be found in the tables and the figures because they may not present visually very well. So make sure you're reading actively. You've got a pen out, or if you're reading digitally, you're using some highlighting function so that you can keep track of things that caught your attention so that when you come back to it again in the future, you can find it again quickly. When you read a table, you're going to have to slow down and spend some time and figure out the layout. Read the title of the table. The actual title of the table is going to tell you something important. Don't just jump right into 45,943. You take data out of context and you're going to get lost or you're going to misinterpret it. So slow down, read the title, look at how the data are organized. What are the headings? And then work your way through those columns, trying to look for patterns and trying to make some sense out of what the information is telling you. The same is true with a figure. A figure is less tabular. It's a little more visual. Often it's in the form of a graph. Here it's in the form of plasmid maps. Before you start digging into the maps, read the figure legend. There may be some key piece in there that's essential to the way you're going to interpret that. Then spend some time, and you're just going to have to slow down. You can see how busy these are, familiarizing yourself with what you're actually seeing, and make sure it makes sense to you before you then jump back into the text and continue reading. An alternative strategy is if you're really only interested in, say, one data set in this whole paper, go to that figure or table before you ever even read the methods and materials or the rest of the results section, and work through that figure, referring back to the text in the results and referring back to the methods and materials only as needed. Only as needed. If really and truly that's the one and only thing you need to get out of this, you can start there and then bounce back and forth to the methods and to the results text in order to make sure you understand that data. Lots of different ways to skin this cat. We'll get into a lot more detail in a later video that's specifically on methods and a separate one specifically on the results.